Well, I see that the number of participants is stabilizing, slowly creeping up, but getting nearly constant. So I will start. So welcome everybody to the next episode of the International Microscopy Lecture Series. It's a joint initiative organized together by the RMS, Canadian Society for Microscopy, Israeli Society for Microscopy, and Brazilian Society for Microscopy and Microanalysis. I am Natalie Resnikov of McGill, and it is my great privilege to introduce today Dr. Harold Hess. Dr. Hess is a senior group leader at the Janelia campus of Howard Hughes Medical Institute. He received his PhD from Princeton University in 1982 in physics, and then accomplished a postdoctoral fellowship at MIT. He worked at Bell Labs, where he developed low-temperature scanning probe microscopes. And in 2005, together with his former colleague, Eric Betzig, they learned about photo-activatable fluorescent proteins and invented a photo-activated localization microscopy that allowed them to break the diffraction limit and visualize the finest details of cell structure. So he eventually returned to electron microscopy, and in his current position is developing 3D techniques for larger volume imaging of entire cells, tissues, and even complete fly brains to map the wiring structure. So today we will hear about larger volume imaging with electrons and cryo-super-resolution microscopy. We will have a question and answer session at the end of the talk. Q&A will not be recorded, so feel free to ask all sorts of questions and also put them into dialog box on Zoom, so that if you have too many questions, we will still recover them, and Dr. Hess will answer them offline. Thank you. The floor is yours, Harold.
Thank you. Yeah, thank you for the kind introduction and the invitation. It'll be a lot of fun and a real privilege to share my path through microscopy through the years. And to start, I'd like to sort of focus on the first word of the title, larger. So I'd like to give a little perspective of where I started and how I eventually sort of ended up into this focus on the larger side, and sort of follow through on some experiments and paths toward which we're going. Now, if you step back, often it's good to really ask basic questions. Why do microscopy? And for me at the beginning, it was just trying to see the fundamental building blocks of the world. And in about 1986, this was sort of a pretty well-established and very noted method of microscopy using scanning tunneling microscopes. And they were able to see this one element, which are atoms, pretty impressively with a very easy apparatus. Most of you are probably familiar with it, but the idea is you scan a metallic tip over another conducting surface, and to achieve constant current, you end up tracing out individual atoms, as in the picture here. Now, at the same time, superconductors, particularly high-temperature superconductors, were becoming very noted, and I decided, well, let's see if we can see some fundamental building blocks of superconductivity, and thought, well, can we see individual magnetic flux quanta, which are characteristics of superconductors, or even the quantum states of parts of superconductors? And that led me to look into scanning Hall probe microscopy, which can measure magnetic fields, or tunneling microscopy. So the idea is you just move a probe across the surface, and you see the variation of either magnetic fields or electronic states, and these can then be imaged, which was, a lot of fun. And there's just another example of what one can do with it. See, these are individual flux lines, the smallest quanta of a magnetic flux line, that can be sort of created. And so progressing onto that, I thought, well, okay, single electrons, let's try that. And one could make a special kind of transistor, use it as a probe. And if you scan it across the surface, you can measure the surface electric charge, and to a resolution that you can even see single electrons. So here's a surface which was scanned, and at the other side, the bright spots are where extra electrons have moved onto compared to the previous surface, and the dark spots are where they have been removed from those areas. So you can see individual single electrons. At the same time, my colleague Eric Betsick at Bell Labs was seeing individual fluorescent molecules, and W. Morner was doing this independently in a different way. And we teamed together at Bell Labs also to look at the individual pieces of luminescence that happens from quantum wells. So there are little pockets or caverns where excitons can get stuck, and you can resolve their spectra and see where they sit locally, as this picture for one specific spectra. Now, the big thing, this was a lot of fun, but we were sort of asked at Bell Labs to think of a larger picture. Where is this going to be relevant? And the trends at the time were really to look at larger numbers. Computer chips were expanding, more and more transistors per chip. Hard disk drives were able to hold ever more data sets, a larger number of bits in there, and the trend was toward large numbers, the mega, the giga, and soon the tera. So at that point, decide, well, let's really look at that, and joined a company here called KLA-Tencor, and this is like a $50 million microscope, essentially, which inspects masks for the semiconductor industry, or could also, different kind of microscopes can inspect chips, and there's a need to do hard disk drive inspection. And it was fun just to immerse oneself in this industry and see all the tools. At the time, they were developing multi-beam scanning electron microscopes, FIB-SEMs, which I'll explain, or maybe you already know, was used for modifying masks. And I think it was useful just to see, the extent and power of how they use these machines. So a little while later, I decided to get involved in another customer-driven problem, and that is to look or help decipher the circuitry of the fly brain. And that's sort of what brought me eventually here to Janelia. And so the challenge at the very beginning seemed to be very analogous to what the semiconductor industry was doing. There, at the time, they might have had a Pentium 4 dual core chip, which had like about 5 square centimeters of area. And if you imagine taking a fly, chopping it up into 70 nanometer sections and spreading it out, you have maybe about 5 square centimeters of fly brain tiled out. The size of the wires, 60 or 50 odd nanometers, and in the fly you have axons and dendrites sort of on the same size scale. You have hundreds of millions of transistors, and the synapse count also sort of goes into the same ballpark. Analogous to nerve cells, you might have computational blocks, which would be the case in the FPGA, where you might have 100,000 of them, comparable to the number of nerve cells. So that all seemed appealing, and so okay, why not? Let's try it. But first we have to think a little bit about it. The volume of a fly brain, you can see, is smaller than a millimeter size scale over here on the right side. It's actually about a 40th of a cubic millimeter. And in order to see the details, you need to sample with about 8 nanometer voxel sizes. And so you just divide that by the volume and you say, oh gosh, you got a good fraction of a petabyte of data, maybe about 1/10 of a petabyte, a little bit more actually. And if you sample at a megahertz, you can quickly calculate that you need years of operation to acquire that data set. And the really constraining thing is that the data needs to be perfect. If you're looking at wires or nerves that go from one side to the other or down here, you don't want to have a break in there, because then it'll be totally, the connectivity is just totally lost. And so there's an extreme demand on perfection and no lost data. So at the time, there are a number of standard ways to do volume electron microscopy. Usually, all of them were based on diamond knives. And the traditional way is you cut sections and image through with a TEM. There are other approaches where you can just cut a surface with a diamond knife and look at that block face, pioneered by Winfried and Ken Hayworth, who you'll meet again a little bit later in the talk, said, oh, let's cut them up, put them on tape. He was working with Jeff Lichtman, and then we image with SEM on that. So we decided to take a little bit of a different approach, which is not diamond knife, but use a focused ion beam to sort of get even finer resolution. And the idea is you scan with an electron microscope and you see the contrast on the backscattered electrons, and then you ablate a tiny layer of a few nanometers. And then you scan again, and then you ablate again. And you keep the cycle going until eventually you sort of ablated your way through the entire sample and you get lots of pictures. So the key distinguishing thing is that with focused ion beam, you get much finer resolution on each one of these steps. If you have diamond knife, you have a little bit a choppier increment, and that can lead to a loss of resolution in one dimension. And particularly for the fine details of the fly, it can make tracing some of these very fine processes difficult or maybe even ambiguous, and could mess up the interpretation of the wiring diagram. So got one of these machines about 10 years ago, or actually more than 10 years ago, maybe 15 years ago, turned it on, and it ran for about 3 days, and then it needs reheat, other problems happened. So with that, we got a mere 1/8,000th of a fly brain. So there's a long way to go. And so the problem is this machine is not that super reliable. It's very finicky. It can last only a few days. And we need to figure out a solution about it. And you don't think too hard, and you say, well, okay, we just have to make it reliable. And so that meant about 10 years of reliability engineering, and fortunately both Sean and Janelle have been patient enough to support that. So we had to tame the focused ion beam, make sure it stays under control and doesn't cut in a bad way, minimize interrupts. If we had to stop, do a graceful restart where we pick up exactly to within a few nanometers of where we left off, and a good maintenance schedule, and a lot of learning from errors. And so here, for example, is one example where the focused ion beam strafes the sample here in yellow, and we measure the beam that goes over the top, and we wanted to monitor that, make sure it stays at just the right level, that only half the beam goes over the top, so it doesn't dig in too much or miss it by too much. And then we have other currents as guardrails to manage and make sure we don't accidentally burn the sample too far. And with that, we had another stab at the fly brain. This time we got quite a bit farther. This represents about 3 months of continuous imaging. This is the optic lobe, and you can see just our beautiful wire bundles going back and forth in different directions. Cell nuclei here in the middle sort of come up and go. There's one right there. And all the dark areas, the big dark things, are mitochondria. These little dark, tinier regions right here are the actual synapses, that define the connectivity between the nerves. But then, to get even more ambitious, to get after the whole fly, we have another problem. This FIB-SEM can't mill very far. If you go beyond 50 microns in depth, it becomes unstable. You begin to have streaks and waves and curtaining. And essentially, the region where you image, you sort of make a burnt crust of material. And when you go in with a gallium beam at a very shallow angle and try to mill it, the milling becomes a little bit unstable. And I like to think of it as a bit like wind coming in from across a pond. The upwind side is relatively calm, but by the time you go pretty far down, the downwind side, you begin to have waves and much more activity. And the milling then becomes a bit non-uniform. So solution was, well, we just chop up the sample. And Ken Hayworth was the one who proposed that and developed something called a hot knife technique, where the fly is chopped into about 30 sections, each 20, 25 microns thick. They get sort of mounted in little tabs like that. And it allows for nice parallelization now. So now we can not have only one sample in a microscope, but it's separated so we can get a lot faster throughput. And the final critical thing is that these tabs and these sections do have good enough quality that you can stitch across that cut interface. So what we've done with that, we've developed, which is effectively a hybrid sectioning and milling. And you'll see this theme come back a little bit later. So we mill with the FIB-SEM at fine increments of about 8 nanometers, but we also use a diamond knife to cut it into 20 micron increments, and that's nicely solves the problem of the limited milling depth and allows us to go after the full fly brain. So a little while later, you sort of come back to life again and try to figure out what happened. This person, Juan Liu, worked very hard on developing the protocol for these flies. He screened hundreds of them, got the dissection and the staining okay. From that, a mere subset of 44 flies were competing for the top position. 43 lost and ended up in this unmarked drawer. And one won, number Z0720-07M. And he is reserved a nice position in a marked drawer ready for FIB-SEMing. This was then given to Wei, who ran the machines, imaged them, and Sean Zhu, who sort of oversaw that to make sure all the machines are sort of operating properly. So there's a room. We have 8 of these machines, and also with the help of Art Crow, technician from Zeiss, really helped everything go. And over a period of a full year, we managed to image the whole thing. And this is some preliminary look at the data where the nerves can now be traced, the sections can be stitched together, and they're being proofread and actively refined, to form a good high quality data set right now as we speak. So from the microscopist side, you say, oh gosh, what happened? And I'd like to sort of think of it on this 2-dimensional scale. The horizontal scale is volume of a sample with about 1 cubic millimeter up here and 1 cubic micron down here. And the vertical scale is resolution, finer resolution being lower down. So typically with the TEM and tomography, you can get good resolution, better resolution than you can with SEM, with scanning electron microscopes. You can stitch a few serial sections together and get slightly larger volumes. But FIB-SEM now seeks to address this little open white space out here. It's a little bit finer resolution than the diamond cut block face approach. Because now we have isotropic pixels, maybe going down to about 8 nanometers, maybe a little bit finer. And the fly brain sort of ends up right here in this this regime. So, okay, so the fly-plane circuitry is on its way, and what more can one do with this? Is there some further value? And I do have a little bit of a customer mindset, so sorry for repeating this here. For the fly, we were operating mainly for throughput. We wanted to go in there and image this thing in 1 year and not 2 years. So we opted for a little bit higher landing energy. This made the beam a little bit broader than if we would have gone for a lower energy, but it did give us better signal to noise. And you can see as a function of landing energy, if you go down in landing energy, the contrast sort of drops a little bit. But you do get better resolution, less depth penetration, less lateral blurring by going down. So if we want to see better, you can drop the energy down a little bit. Another thing that one can do is we were operating with higher current. And higher current in the SEM means, well, a slightly larger aperture, but the magnetic lenses are aberrating. And so if you go to a smaller aperture, you get less chromatic, less astigmatism, and you can get a little bit better resolution. Now, the big downside by cutting down the aperture, it means way less current, but also considerably less, considerably less throughput. So what does that mean physically? That means the images can go from something like this, which is what we had when we were trying to image the fly brain, to something that looks like that. But the volume throughput can be significantly lower. And we gain a modest increase in the resolution, but nothing nearly as dramatic as the lower throughput. But anyway, this still puts us in a very interesting regime. So one of the first things we decided, let's just spend a full month imaging one HeLa cell at this higher resolution. And here's just a very tiny part of it, just to get a sense of the detail that you can see. Again, not as good as a TEM, but we got the volume. There's Golgi out here. There's a microtubule that's going right across here. You can sort of barely make out the hollow structure in there. Here's a nuclear pore. This is a nucleus inside here. The let's see, the ribosomes are up here. Nucleosomes might be these little spots that you see down inside. And but this is now just of a small area. That little small area was annotated by a group called COSEM at the time. It's now reformed into something called CellMap at Janelia. And this can be done just for a few cubic microns, very, very directly. And now using that data, that can sort of form the ground truth to which you can sort of begin to do some machine learning. Here you can see the microtubules in here, all the other structures, nicely labeled. And now we can sort of go apply that to a whole HeLa cell, and you can sort of tease apart all the different parts, the mitochondria in orange, the nuclear membrane in purple, ER in green, and so forth. And this is actually a very easy one-button kind of machine that can just, I think now, just generate these volume stacks for just a variety of experiments. So one little collaboration here with Harvard, where we could image liver under different metabolic conditions, high or low glucose, and you can see the structure of the ER in the whole cell, whole tissue context, and compare it. Oops, too far. And for example, between an obese and a lean, you can see just sort of very, very different structure between the ER in one case and in the other. So the big thing is with the FIB-SEM, it allows you to characterize cells not just based on a thin section, but now on a whole cell basis. We can count all of the nuclear pores. We can sort of find all of the ribosomes on the outside. We can sort of do a full characterization of all of the ER, and potentially ask a lot of questions which were sort of, which are otherwise a little bit outside of the range of when you just take a single section view of the world. So if you want to just sort of get a little bit of sense, CellMap opened up a website called openorganelle.com, and I'd encourage you just sort of find it. It has a lot of data sets. You can click on it. It's a NeuroGlancer link, and you can simply browse the data. Some of it has been segmented, and you can see it in 3D. A lot of it has not been segmented and is available for download or further analysis. And we have different tissues, macrophages, kidney cells, or kidney tissue, and the like. And we're hoping to sort of continues to add on this and maybe sort of get a first stab at what particular tissues might look like. But then, of course, they're going to be in-depth experiments for the biologists to really harness and mine what's really for their particular problem. So, yeah, so again, so the whole idea is with FIB-SEM, we can sort of go from, instead of a characterization just based on sections, diamond cut sections, to whole cell biology. And essentially now this is exploring this other part of the phase space, a little bit smaller volume, a little bit better resolution, so it's a little bit down to the left and right. So one other direction to go with is to note that the electron microscopes really give great global context. You know, the membranes are stained, certain protein complexes are stained, and it provides a lot of information. On the other end, fluorescents usually end up labeling 1 or 2 or 3 specific target proteins out of 10,000s. So you're really sometimes lacking a bit of context. You know, you might see, if it's a relatively sparse protein, you just sort of see blobs floating in space, and it'd be good to put context in there. So the two techniques naturally can sort of go together to form a correlative approach. Initially, this was the kind of world that one was going back and forth with between electron microscopy, where you have good, strong fixation and you can see details, or you do poor fixation and you lose it. But when you lose the details, you preserve a lot of the antigenicity or the fluorescent properties that you can get good super-resolution data. So you're working between this bad-good or good-bad situation. Fortunately, there's another approach, and what's nice, some of the techniques in electron microscopy have already working with cryofixation, and so with cryofluorescence, one can maybe get past this little link. So the normal workflow for the FIB-SEMs is you culture cells, let's say on a cover glass, you high pressure freeze, and then you do freeze substitution, pop it off the cover glass, trim it a little bit, put it into the FIB-SEM, and then you see all the osmium stained regions of the cell. But if we stop at this point and insert transferred into a cryogenic microscope, optical microscope, for example, right here, we just moved the sample in, presented it to the objective. We can further send that light over into structure illumination microscopy optics or a palm, single molecule resolution kind of stuff. And it works. A super nice thing at low temperatures, these fluorescent molecules are pretty much very robust. They don't bleach easily, so it allows us to really get full 3D structures in SIM very nicely without the first layer being strong and then the last layer being sort of bleached out, so we can get the entirety of the cell. The molecules at low temperature are also blink, or certain subset of them blink. And you can then use the single molecule localization on them. It's much slower. It takes us, hours, days to sort of get a nice data set. So it's not the fastest thing in the world. But anyway, it allows a method for getting correlation. So now we just insert this little extra step in the process, and the same sample gets a correlative image. So we match the FIB-SEM image over with either a SIM image or a palm image, and you get the two overlaps. The SIMs are quick, great for overview, but the palms give you a little bit more detail. And of course, they have to be registered. If you look carefully in, the features in the optical microscope might not align with the features in the EM. Usually they can be identifiable depending on what you label. If you label something which is very common in the sample, then you have a number of references to remap it. And because of the final processing, the dehydration and the infiltration, you do get some distortion, and you do have to make a mapping, the landmarks from the optic side to the EM side, and you can compensate for all of the distortion and get a good overlay using this big warp software that Stefan Saalfeld sort of pioneered. Here's some examples. Here's ER on the optical side, in yellow, mitochondria and cyan, corresponding EM images, and they overlay nicely. Now, sometimes you might just see a tiny little spot in this yellow color associated with the ER protein, and you think, well, maybe that's just a piece of junk or nothing relevant, or same for the halo, the TOM20 tag. But if you look at the EM image, you see, well, there actually was a tiny little vesicle there. So what that's saying is that you can actually identify the different kinds of vesicles by their protein content and separate what's otherwise maybe indistinguishable under the EM. And there's some more games, proxysomes. You can label them, identify what kind of little structure that is in EM, label it, do it for another one, label it, and work your way through the whole cell. And you can systematically say, oh, this is all of the proxysomes have this size and shape distribution, and get a comprehensive whole cell kind of sense of what proxysomes look like for that particular cell. And another example, this is some work that Alex Ritter did with Eric Betzik earlier, where you can see T cells attacking a cancer cells. The red are the little lytic granules, which will later get presented to the cancer cells. And so that cover glass of those cells can get frozen, high pressure frozen, and you freeze them in the act of whatever they're doing. And we can then select one of them optically from that 3 millimeter disc, and that can sort of be pulled out, trimmed with FIB-SEM, and you can actually then see this immunological synapse that's forming between the T cell as it's attacking the cancer cell. And from the side view, you can see this actually sort of cups in as it grabs the T cell, right there. The dark spots here, those are lytic granules, which will then break the membrane of this cancer cell, this HeLa cell. And here's the immunological synapse, where essentially the big biological warfare is taking place. The cell is trying to put up a bunch of actin here, trying to shed membranes, and you just see the full activity going on. So just to summarize that quickly, both SIM and PALM work at cryogenic temperatures. Cryo-SIM is less bleaching, so you can get better Z-stacks. PALM works nicely, but we do have vacuum gaps with the objective, so we have to work with a lower NA than we do at room temperature, but we got way more photons to deal with, so that compensates for this lower NA. We like the cryofixation that you have when you're using this particular approach. And there's a rough sum numbers of what the resolution is with Cryo-SIM or with Cryo-PALM, and then with the FIB-SEM, a bit of a time scale to collect the data. So what we've done here is on this, we've just added some protein-specific colors, not with the full FIB-SEM resolution, but enough to be informative. Now, here's one final thing that I just want to point out. We can go to yet even bigger volumes using something called the Multi-SEM. So what could we do in the future? Can we get larger volumes? So for larger volumes, we need more beams. The beams are essentially limited to the order of a nanoamp or a fraction of a nanoamp just by space charge. You just can't put too many electrons into one beam before they begin to sort of space charge broaden or suffer aberrations or other things like that. But more beams can be done. So one solution, if you want 100 times the throughput of one machine, so you just get 11 more rooms of the same thing and you're there. Well, let's see, again, thanks to the demands of the semiconductor industry, number of approaches have been put forward to develop multi-beam machines, and Zeiss has one of these multi-beam machines, which right now is being used in biology. The idea is you have a source, several beams, it all gets focused down onto this sample, and you have this little beam splitter, this little magnetic thing, which then sends the resulting beams back out onto a detector where you detect each one of these channels. And this is what it looks like. You just buy it for a lot of money. And over here, you see one little section, which corresponds to one beam. It is imaged here, that they're tiled together. Here are 91 beams, all tiled together. And so each beam is scanned together with all of the other beams, and you collect this hexagon-looking pattern. And then you move the sample over by about 150 microns, and you collect the next hexagon, and you just sort of continue it. Now, the one problem is you need a compatible way to mill it. You can't use the focused ion beam. But we can come back to this idea of hybrid cutting. So we can cut with a diamond knife 1 micron thick sections and then mill it. But we need a different milling technique. So Ken Hayworth originally suggested do a gas cluster ion beam milling, where we cut these 1 micron thick sections onto a plate. The gas clusters provide a gentle milling action, and it does seem to work. So essentially, we image each one of these sections, mill it, image, mill it, and each time we go through this image mill cycle, we generate another plane. And we have different stacks for different sections. All of these can then be flattened. They don't mill through exactly at the same time, so the software is needed to register and flatten and stitch them back together. And it does look very compelling in the end. And let's see, yeah. And so here's an example where one was actually able to trace neurons through such a data stack. Thomas Templier was trying to do pretty much the same thing out in Switzerland, but he was using argon atoms, but not in a gas cluster, but at a way shallower angle, and also has very interesting, results. Now, at Genili, we put these two ideas together, and we call it the IBEAM-SEM. So we mill with either gas cluster or ions, and then we add one extra step. We do a little bit of an etch. The etch is important because it prepares the surface to make more secondaries and give a better signal to noise on the contrast for the sample down below. And this is what the machine looks like. We buy this and we put this together. And let's see, I'll see if I can get this to go. yeah, so here's a robotic. This all has to be in vacuum chamber. So first we make 2 wafers, cut the sections on them. We first have to burn them to make them conductive so that they could be imaged. Here's a multi-beam coming down, imaging one set, and then another and another. And then we go in and we mill it with the beams. And at the same time, we're imaging over here while we're milling it over here. So we actually have 2 wafers and we swap. Now this is going off into, oh no, sorry. I stopped it. Yeah, so it's etching back there. And after it's etched, that'll be presented for the imaging, little robotic arm, while the other one is being milled. And this continues about, maybe a day to go through all of it. And eventually we collect these stack of sections and put them together. And this is what it looks like after we really put it together. And we're just running the first samples. Here's an early sample. And here's a hexagonal pattern of multi-beams. It's first optically imaged to find them, to make order. Here we have 19 areas. We're just looking at the middle. And here's an example where we look at it. As you can sort of see, each little flash as we're going through one layer, but they stitch nicely. Side view of the same thing. And this data set can then be given off to our friends at Google. Mihal there sort of can segment out the different parts, and the quality is good enough that you can find, synapses, click on them. And the nice thing is they are continuous across the whole volume. We don't see any breaks or fragments, which is very, very promising. So, so what's next? I think next we're actually very hopeful that we'll be able to get a female fly. This will take about 4 months. We've been, at the edge of launching this, but not quite yet. These machines are pretty agnostic. They can look at anything, and their discussions are all over the place, whether it's fish, spinal cord, other mouse brain pieces, tissue, organoids, plants, little critters, whatever. And we've, I think, yet to still fully look at, okay, what are the real limitations on the throughput, and that's going to be limited both by the imaging and how quickly we can step and settle with the stage, still to be explored. But this gives us a possibility with these machines of imaging either 2 or 8 cubic millimeters a year, something of that range. So, okay, there are a lot of people who are crazy ambitious and they want to image something on the order of close to a cubic cc, like a mouse brain. So what is that? That puts us maybe all the way out here, well away from this space. Well, how do we get there? Well, for the mere price of a 767, we can buy maybe about 20 of these machines, have them run for 5 years, and you could almost mentally imagine that something like this could be in reach as a target. So, but this is beyond what certainly we'll be doing here. This will require a bigger effort, and a lot of people are sort of actively pursuing that. So just a final thought that I'd like to take up, and that is, again, sort of drawing from what these folks, our friends in semiconductor or in the industry side, what they do. I'd like to think of their machines as actually difference machines. With these machines, you're just generating tremendous amounts of data, but not all of it is quite relevant. So what they do is they image one part of a sample, and then for the next sample, they image the same part. And then you can just simply subtract the two images, or this might be a database. And you subtract the two images, and what's most relevant comes up, and which is a defect, maybe a broken wire or a short or things like that. And so these are basically difference machines. So in the future, I think, and biologists do like to look at differences. What's one mutant compared to another mutant? Or here's a wild type fly plus a not so wild type fly. We essentially have to make the same idea of a difference operation. This is not trivial for biology, and I think it's one of the big, I think, really cool open challenges as we try to think, well, as we get more and more samples, learning how to sort of extract and tease out the biology will be a huge challenge. And with that, I'd just like to thank you for your attention.